Good morning. Good morning. My name is Ononiwo Emmanuel Chidera, and I'm speaking on Jak-Stat as a signaling and its therapeutic relevance. Okay, Jak-Stat signaling, it's a way for the cells to respond to cytokines, which are molecules that regulate inflammation, immunity, and other cellular processes. Cytokinesis bind to receptors on the cell surfaces, which are made up of two amide chains. When this cytokinesis binds, it causes the receptor to come close. This is a phenomenon known as receptor decimalization to form a pair. They come together to form a pair, and this process brings JAKs in close proximity and allow them to phosphorylate each other as well as the receptors. Now, after this phosphorylation, it creates a docking site for proteins known as STATs, which has a special domain known as the SH2 domain. Here, as you can see on the screen, Jak phosphorylates STATs, and then now the phosphorylated STATs disassociated from the receptors form what we call dimes with other STATs, and then these dimes start forming pairs and move to the nucleus to bind with the specific DNA found in the nucleus, where in a DNA sequence where it turns on and off some certain gene expressions. And yeah, you can see this is the downscreen, an example of how it passes through. And then yeah, there are some canonical mechanisms that I mentioned. Non-canonical phosphorylates histone and exclusion of chromatin openings. See, you can see how STAT cooperates with cells to integrate signaling. Okay, this is another signal here that shows the negative regulation and feedback inhibition, where how the pairing and the phosphorylation goes down to the gene expressions and how it's being phosphorylated. That's the P, the STAT. This is the coming together of the JAK. There are some genetic diseases and mutations that comes with JAK pathway, which is listed here, like leukemia and all that. Okay, therapeutic targeting. These JAK they have JAK inhibitors. One of their defects is selective iso. Okay, this is genetic how to study JAK receptors, genetic knockout on mice, and then ICP4 receptors, JAK association for kinase assays. The next one is a functional reporter assay. Okay, this is quite self-explanatory here. Okay, this is another visual representation of the pathway where the tyrosines and then the coming together of the JAK, the phosphorylation, the separate STATs when they come together and when they form a pair before they migrate into the nucleus of the cell. These are some importance we'll take home from the cellular response of dozens of growth factors are mediated in the pathway, transphosphorylation, and then downstream substrate. Okay, activated STATs enter the nucleus to bind the sequence to the target gene. JAK mutation cause numerous diseases. Okay, I think this is one of the keys to take home. These are the reference selected. Thank you.